Your body constantly needs new cells. In fact, right now, your red blood cells are dividing in order to produce 200 billion new cells each day. And during this process, your DNA, which is organized onto chromosomes, is copied and then distributed to the dividing cells. Now, one of the worst things that can happen during all of this is for a cell to undergo division with a broken chromosome. So why is this bad? Well, think of it this way. Cell division is kind of like Xeroxing a stack of blueprints and handing those stacks out to two sets of construction workers. Now, when you have a broken chromosome, that's kind of like dropping a page or two out of your blueprints before you hand them out. So when the workers go to build their buildings, there's going to be something missing. Maybe the elevators, maybe the restrooms, maybe something more important. And in the case of cell division, these missing parts can often lead to the death of the cell or to that cell becoming cancerous. For example, the loss of broken chromosomes in division is believed to play a role in the development and progression of colorectal cancer. So there are some really important medical implications if you can understand how a cell may deal with a broken chromosome. Or in other words, can you find a way to return those dropped pages back to their proper stacks? To study this issue, I use a technique in which I can regularly generate broken chromosomes in the developing fruit fly brain, and then use cutting-edge microscopy in order to monitor their behavior during division. And what we see is, surprisingly, these broken chromosomes are actually able to rejoin the intact DNA in the final steps of division, allowing the break to be repaired and the cell to survive healthily. And in my research, I've identified and characterized one mechanism for how this occurs. So normally, your DNA is surrounded by a structure called the nuclear envelope. Think of the nuclear envelope as a wall that surrounds a castle. And while this wall is important for protecting your DNA against any potential foreign invaders that may try to damage it, it's also problematic, as you would expect it to act as a barrier that would block a broken chromosome from rejoining the DNA at the end of division. Now, what I found is that broken chromosomes are able to overcome this barrier and pass through the nuclear envelope through channels. And so if the nuclear envelope is a castle wall, these channels are the drawbridges and entryways that the broken chromosome travels through in order to enter the castle. And furthermore, what I've seen is that in cells that are unable to form channels, or when the drawbridge is up and the entryway is closed, broken chromosomes are locked out, and you can end up losing or severely damaging the DNA that's contained on the broken chromosomes. With this in mind, I'm currently using a combination of genetic and chemical techniques in order to identify the factors responsible for channel formation. And I'm hopeful that my research may uncover potential strategies for designing future therapies against cancer. Thank you very much.